All right, well, I think that we can go ahead and get started here. Hello, my name is Sabrina Sholts, and I'm a biological anthropologist and the curator of the exhibit Outbreak, Epidemics in a Connected World at the Smithsonian's National Museum of Natural History, where I wish I could be today, but I am joining you from my home in Washington, D.C. On behalf of the museum, I am very pleased to welcome you to today's program, From the Laboratory to a Shot in the Arm. This is our third program of 4 in the series, Vaccines in the Time of COVID-19. The final program in this series will be same time, same place next week, Tuesday at 12:30 p.m. Eastern Time. And I hope that you can join us for the conclusion of what has been a really exciting and informative series. For more than 2 years, the Outbreak exhibit and its associated programming has been educating the public about viruses that spill over from animals and pandemic threats and One Health. I take every opportunity to thank the many organizations that have made that possible and supported us all along the way, including those that have provided us with funding. In today's program, we will hear from an expert who looks for transformative solutions to global health and development of vaccines. But before we begin, I'd like to acknowledge current events and let you know that the National Museum of Natural History stands with the Secretary of the Smithsonian, Dr. Lonnie Bunch, in expressing our deepest sympathy to the families and communities impacted by discrimination and violence. We'll be sharing Secretary Bunch's response to these events, as well as a resource developed by our colleagues at the Smithsonian's National Museum of African American History and Culture. Talking About Race is designed to provide tools and guidance to inspire conversation about race and empower your journey. So we'll share those links with you in the Q&A. So note that the Q&A box is where we'll be communicating with you throughout the program. We'll post other links as they come up in our discussion, and that's where you can submit your questions for our speakers. So please take a moment to locate the Q&A box, which should be either at the top or bottom of your screen. You'll type your questions in there, and we'll do our best to answer as many of them as we can. So now to actually get started with our program, I would like to introduce you to our speaker, Dr. Lynda Stuart. Dr. Stuart is Deputy Director of Vaccines and Human Immunobiology at the Bill and Melinda Gates Foundation. In this role, she leads a team that aims to source novel approaches and accelerate the discovery, development, and translation of new and active immunization strategies for diseases such as malaria, HIV, and of course, COVID-19. Prior to joining the foundation in 2016, Dr. Stuart was a member of the faculty at Massachusetts General Hospital and Harvard Medical School, where she was co-director of the Laboratory of Developmental Immunology. She was also a member of the Center for Computational and Integrated Biology, an affiliate of the Broad Institute of Harvard and MIT, and served on Massachusetts General Hospital Executive Committee for Research. She remains actively involved in basic research somehow. And we're so thankful for what she does and giving us some of her time to join this program today. Lynda, thank you so much. Welcome, and please take it away.
Thank you, Sabrina and team, for the kind invitation to give this talk to the Smithsonian webinar series on outbreaks and epidemics in a connected world. I will go ahead and share my screen. So, as Sabrina introduced, I work at the Bill and Melinda Gates Foundation, and I'm presenting today really our perspective on the COVID-19 vaccine response and this journey of trying to get from the laboratory to the arm of patients in unprecedented time. And I'm presenting on behalf of the COVID vaccine response team. Sorry, I'm having a problem with my computer screen. Hold on one second.
Yeah.
Sorry, I have a problem with advancing my screen. Hang on a second, I will. Okay.
Okay, great.
Sorry about that. So the Bill and Melinda Gates Foundation started our COVID vaccine response in January with 4 main goals. Those goals are listed, are shown here. The first was to protect the most vulnerable by rapidly strengthening capacity and coordination to detect and respond to COVID in key countries, particularly those in low and middle income countries. To accelerate the detection and containment of the virus by filling gaps and informing global response through partners with the capacity for rapid action. To develop treatments and a vaccine to coordinate and fill gaps in COVID product development to secure equitable global access. And to minimize societal impact and economic impact of this devastating epidemic. Our R&D, our approach has been really threefold. First, to identify and develop diagnostics to test for COVID vaccine cases, to look for new treatments by identifying and developing therapies to help people who are infected with the virus to recover, and also to prevent by identifying the development of vaccines that could prevent the transmission in the first place. And these 3 areas of function as a continuum, even if a safe and effective vaccine is developed, treatment and testing will be essential, very much like other diseases such as influenza. At the foundation, vaccines are a priority. We are very heavily invested in vaccines as they are the public health tool with the greatest impact. Over the past 20 years, the foundation has spent billions of dollars on developing and deploying vaccines to protect children in low and middle income countries. And every year, the discovery team that I work on at the foundation spends $50 million on tools needed for the discovery and translation of vaccines and monoclonal antibody therapies for global health indications. And we do this in a variety of ways. But in addition to our disease-specific efforts, we invest in disease-agnostic way and have built a comprehensive toolkit for vaccine and antibody R&D. We've built an enabling framework and a large network of partners that can support vaccines for any and all foundation diseases. And we prioritize cutting-edge platform approaches over disease-specific solutions. And we bet on novel technologies and innovative solutions. And we're very comfortable in working on programs with long timelines, such as the mRNA vaccines that you've seen a lot in the news, which we first invested in 2013. This really has brought us together, allowed us to build a toolkit that is able to support many sorts of vaccines, subunit vaccines, nucleic acid vaccines, and viral vectors that are all now being leveraged for COVID-19, antibody therapy, correlates, and we also work in the livestock vaccine area because we realize that livestock are both essential for supporting the livelihood of people in low and middle income countries, but also because of the One Health threats around spillover from zoonotic diseases. So supporting all of this work, we have also a network of global health vaccine accelerator platforms that both support basic science and provide an enabling framework. We'll talk a little bit more about that today. Our existing foundation investments have enabled a rapid response to COVID-19. So we've worked in partnership with the Coalition for Epidemic and Preparedness and Innovation, otherwise known as CEPI. CEPI's early response was focused primarily on speed. But the foundation, realizing that we were really in a global pandemic, launched a vaccine program, a discovery program within about 30 days of the identification of SARS-CoV-2, with the goal of enabling the field in general and our partners such as CEPI specifically, but to develop a portfolio of wave 2 vaccine candidates that were focused on affordable and scalable solutions that could be deployed in low and middle income countries and as a global pandemic response. So how we have done that was really to do 6 things. So the first was to invest in understanding the basic science of the virus. The second was to use our existing vaccine platforms, which we could deploy rapidly. The third was to leverage our network to build an enabling framework. Fourth, prioritize vaccines that could be made at a 1 billion dose scale. And what we are now looking at is how we can accelerate the development and then the deployment of these vaccines through partnerships. Before I go on, I think it's important to understand what we are trying to do and why it's unprecedented. So normal vaccine research and development takes about 15 years. So if we were to extrapolate that from where we are today, we wouldn't have a vaccine until 2035 in normal circumstances. And that's because vaccine development requires all of these stages: phase 1, discovery through phase 1, 2, 3, and registration, which normally happen in series. What we are seeking to do in COVID-19 is to concatenate that response and to try and develop a vaccine in unprecedented speed in less than 2 years, and we're trying to deploy these vaccines in less than 3. So what are the main features of these different phases of development? How are we doing things differently in COVID-19? And who are the global players that are all working together to try and make this happen? So I'll focus first on discovery and preclinical development. And I'll spend a bit of time here because this is where we've actually already shortened the timeline quite significantly. So vaccine development is usually very hard. The probability of success of a vaccine is about, in these early stages, only 7%. And if it gets to phase 1, it's about 17%. So what are we doing differently? We're using different platforms. We're building a portfolio of candidates, both simultaneously and rapidly. And there are multiple players that are helping to make this happen. The Gates Foundation, the NIH, previous investments of the foundation and CEPI and platform approaches are being used. We've established centralized labs, and the NIH and CEPI have funded the early development of candidates that they hope to move into phase 1 by the end of the year. And the foundation is supporting a portfolio of wave 2 vaccines that we hope can be manufactured at scale. So talking a little bit about the basics. So the first thing that we've made significant investments in is the biology of SARS-CoV-2. So many coronavirus vaccines target the spike protein on the virus. This is the thing there's often a cartoon that you see, which is a gray ball with red spikes. These red spikes are what we target to do the vaccine. Many coronavirus vaccines use a trick that was identified by Barney Graham and team at the NIH in 2017 to stabilize the spike of the MERS and SARS-1. What we've done now is quite unprecedented in speed. So the novel coronavirus sequence released on January 10th, and SARS-CoV-2 spike was crystal structure was rapidly solved by February 15 and published in the open access journal bioRxiv. And I think bioRxiv deserves a specific call out during this epidemic because the rapid sharing of data through this publication mechanism has really enabled the field. So since that discovery in February 15, multiple important basic findings have been identified. We now know how the virus binds and enters cells. And we now have improved spike immunogens that would, in normal circumstances, would have taken years to come about. The other important basic science effort has been to identify antibodies and define how they protect. So most vaccines are designed to drive neutralizing antibodies. And these antibodies can be used as tools to help identify sites of vulnerability in the virus. By May of this year, protective antibodies had already been isolated from patients, including the WA1 patient, who was the first patient in Seattle. And we had already shown that these antibodies could mediate protection when given to animals. These new antibody tools are coming online to aid new vaccine design, but importantly, antibody therapies are already in the clinic. And on June 1st, Lilly began the first clinical study of a monoclonal antibody treatment. The other thing we have done in this discovery phase is to use vaccine platforms. So one of them that we are particularly supportive of at the Foundation are nanoparticles. The viral spike protein is sufficient to induce a protective response. But the addition of use of synthetically designed nanoparticles can allow these spike proteins to be displayed on the surface in such a way that it looks like the virus itself. And that's shown here on the right. The monomeric protein is RBD receptor binding domain on the left, and then when displayed on a nanoparticle on the right. And this displayed protein is much more immunogenic and can allow us to use much lower doses, which may be very important when we're trying to make vaccines at a billion dose scale. For such proteins to work, we require adjuvants to boost the immune response. These are immune stimulants. And we are ensuring that there is commercial access, there is access to commercial adjuvants that have been in people. And we're also working on de-risking new adjuvants that can increase the supply. The other platforms that have been used and have been talked about extensively are the messenger RNA. So instead of introducing the protein, as I showed in the prior slide, here the messenger RNA is introduced, and the body's own machinery makes the vaccine. BMGF has been invested in nucleic acid vaccines such as these since 2014. And the reason for that is because they can be rapid response vaccines. They're very versatile, and they're potentially a disruptive technology. However, it's important to note that none of these have been in a licensed product. We don't know the dose, and we don't know if we can make them at the millions and billion dose scale. This is all things that we now need to understand. But multiple mRNA vaccine candidates are already in the clinic, and additional ones will follow. The other important thing has been to build enabling tools. So we've brought together the animal models. So vaccines must be tested in animals before they can go into people. We often use mice as the first test and see if they induce an immune response. But animals do not have the same receptor as humans, so cannot be easily infected. So for small animal studies, we have also had to develop transgenic animals that are engineered to express human receptors, or we have also discovered that the use of the Syrian golden hamster, which has a receptor, this ACE2 receptor, that is closely homologous to the human, allows it to be infected by this human virus. Non-human primates are also important for efficacy testing and safety. The other tools we are building are assays, reagents, all of these trying to help us understand how vaccines protect. So we know that the assays need to be very robust. They need to support the regulatory filings for and be qualified and validated for use with clinical testing. And assays are also required to evaluate antibody therapies. In addition to this, there are many other tools that are needed to develop a vaccine, and we are working together with these partners of our Global Health Vaccine Accelerator platforms to bring online new and novel assays and reagents that can be used. A really important goal for the field, I think, is the development of a correlate of protection that might allow us early vaccines to enable the fast followers in the portfolio, and maybe at a portfolio level improve how fast we can move the field forward. The other important thing in this very early stage of the R&D process is to really consider how are we going to make billions of doses. And so we have focused on designing vaccines that can be easily manufactured. So we know that 2 to 4 billion doses will be needed. And we've invested in protein production systems such as Pichia pastoris, which is a yeast that can make lots of secreted protein. We also work in collaboration with developing country manufacturers, such as the Serum Institute of India, which is the world's largest vaccine manufacturer. And we hope to be able to leverage their existing capacity to rapidly make these vaccines available. We are therefore designing vaccines that can specifically be manufactured on these platforms or by our DCVM partners. So coming back to this problem that we face of how do we accelerate this research and development timeline. I've explained to you maybe what we've done to shorten this first block of discovery and preclinical development from 1 to 5 years to less than 6 months, but what can we do with the rest of the timeline? So I think to do that, to explain that, I need to first explain what are the key features of these different phases and what are we doing differently and who is doing what. So a phase 1 study is normally a safety study where a small number of individuals are evaluated. For COVID-19, we're conducting preclinical animal testing in parallel to these phase 1 studies. We're also doing these in expanded age ranges, hoping to capture the elderly, who are going to be important target population. We want to show that these vaccines are safe and they can move ahead into the other phases, and multiple phase 1 studies are already underway. The next phase is the phase 2 and 3 studies. These are studies where the phase 2, where we're looking at different doses and dose regimens. We're trying to get into the target population, such as the elderly, and we're looking for additional safety. And phase 3 are the definitive studies that confidently describe safety and statistically show the vaccine is efficacious. These are the studies that we're all waiting for because these are the ones that allow the vaccine to be used. So what are we doing differently here? Developers are opting to blend phase 2 and phase 3 together once they know things are safe in phase 1. We're actually doing this as a portfolio acceleration, where we're hoping that the second wave of vaccines might benefit and learn from the correlates of protection identified in the first wave. But a major risk to this is that the COVID-19 epidemiology is very unpredictable. So phase 3 efficacy studies will require multiple sites, and this is very complex and hard to run. Multiple players are helping here. I think the NIH has a harmonized protocol for the warp speed vaccines, for the BARDA-funded vaccines, and also the WHO are looking to build a Solidarity 3 protocol that may also help pull the other vaccines through this process. The next step is registration and licensure. So if this basically, every vaccine has to go through a review by a regulatory authority before it's okayed for use. We anticipate that these products will fall under what we call accelerated or emergency use, and that to allow them to be used rapidly. So for COVID-19, what are we doing differently? The regulatory agencies are all very aware of the urgency and are doing very quick reviews, but they will not compromise safety because safety of a vaccine is paramount. Each country will have to make its own decision to use a vaccine based on benefit-risk analysis, but on limited data. And post-launch safety surveillance will be required. And who are the key players here? So the US FDA and the European EMA are both regulatory authorities with extensive experience of approving vaccines. And the WHO will also play a role in pre-qualification, but we're not sure whether whether pre-qualification will be the path or whether low and middle income countries will use instead the stringent national regulatory authorities. And then the final part of this clinical development is really around manufacturing. So first, the vaccine has to go through what's called process development, which refines how the vaccine is made and then scaling up for final commercial use. Globally distributed manufacturing will be needed to maximize production. So not only will you make it in one site, but we will scale out to multiple sites. And that will be what will be required to reach the billion dose scale. And this tech transfer to multiple sites can take between 6 months to 2 years to ensure the quality and consistency. This is typically the most significant delays in any vaccine program. How are we doing things differently? So we know we need huge capacity to make the 2 to 4 billion doses, and this will necessitate production in multiple sites. Instead of building new sites, we're trying to retrofit existing sites, as we know this is faster. But there are very few sites that are appropriate and have the type of idle capacity that we need. Scale out typically occurs only after vaccine works. So we are now having to make large investments in manufacturing at risk. And who are the global players and partners? A number of donors, both sovereign, multinational, philanthropic donors, will be required to cover the costs, as this will take multiple $billion to achieve the global scale needed. So global cooperation is absolutely essential to make this possible. The final part of the problem is really this one, is once the vaccine has been made, who will get it and how will we get it out to the people who need it most? For this purpose, the world has come together around something called ACT, the Access to COVID-19 Tools Accelerator, which will focus on both the R&D and manufacturing, as I've discussed, but also delivering vaccines. There are 3 pillars of the ACT Accelerator. The first is vaccine, focused on vaccine partnerships. The second, therapeutic partnerships, and third, diagnostics. But I will focus here on talking about the vaccine partnerships. So CEPI, the Coalition for Epidemic Preparedness and Innovation, will co-convene with Gavi, the Global Alliance for Vaccine Introduction, and together they will be looking at the end-to-end, the R&D and manufacturing by CEPI, and the procurement and deployment of those vaccines by Gavi. This is indeed a global collaborative effort, and this is what will be required to get these billions of doses out to the people who need them in a time frame that is unprecedented. So with that, I'll stop and thank Sabrina and for the invitation to give this talk and take any questions.
Lynda, thank you so much for that. That was fantastic, so impressive. Really a bit overwhelming to realize all that's going into this, that's so critical, and the role that people like you are playing in so much coordination, development, and translation of the science. And at a time where it's a bit difficult in some places, in some moments, I think, to find hope, sort of positive signals in what's going on. I think that the science, at least, is something that we can focus on and talk about. And particularly for me, it's the science that's happening globally. I appreciate your comments about the need for global cooperation. That's certainly a major theme of our exhibit and a message, of outbreak more broadly, that we have to work together, and these are global challenges and problems that we have to face together. So my question is, in terms of trying to work together internationally, is that going as well as it seems to be, or is it should be going? Given that, maybe there's some concerns right now that what should be collaboration might lead to competition among labs or countries when it comes to a vaccine.
So maybe I'll talk first about the scientists, because I think this is where we have seen unprecedented collaboration. The people's willingness to share data in real time is extraordinary. And the basic science and the use of open access journals and sharing of reagents and tools, and I've not seen science done with this level of urgency in my life. And it's moving, right? So to really see people come together to try and help mankind. I would love to see it happen more frequently, and maybe that's the lesson that we will all take away from this, is that, you know, there is an urgency for science to be made into reality. Mm-hmm. So I think the scientists are doing a fantastic job. The level of cooperation at the sort of other sort of global and country levels is very complex. And I think we all need to recognize that there is a balance for any country or sovereign country in protecting their own citizens, and also recognizing that we're all global citizens and that we all really need to contribute to protecting the world. And I think it's this balance is really hard to think about, but I think it's really important when considering a global pandemic, that no country is actually safe until every country is safe. Because you can do everything you like to try and prevent, you know, COVID-19 from, or any other pandemic situation, right, from spreading from one country to another. But if you don't actually address in the, you know, the disease in the poorest countries, they'll still spill over into high and middle income countries. So I do think that people should step back and actually take that perspective as they look at collaboration, that it really is important to get vaccine out to everybody. But I think on the R&D front, actually, there has been a significant collaboration, which we didn't see before in, and I think CEPI has been really important. That CEPI was stood up after the Ebola epidemic, really to try and serve as a collaborating forcing function, and I think they've done a really great job at bringing that together, you know, we will continue to see, I think, great collaboration, and I'm hoping increasing collaboration as we move forward.
If you could say that there would be any particular game changer, or I guess maybe most impactful change that we could see in the way that we're moving right now. It was really interesting for you to point out the spots in this long process where you might see the most delay, or where you're really investing the most or taking the greatest risk for something that you can't know will work out. Is it money? Is it a scientific breakthrough? Is it just boots on the ground? Are there any, I guess, factors that you particularly pull out that we need even more of to even work better or faster towards this?
It's not entirely money, although there are parts of it where it is. Money does play a significant role. I think there are transformations and changes across that whole continuum that I showed. I think the use of these very rapid response platforms that are very agile, if we can actually show that they work and they're safe, I think this will change the field for the future.
Yeah.
I think how we are thinking about, and that's just and there are additional scientific breakthroughs that are emerging every day, right? You know, the speed that we're discovering antibodies, the speed that we're able to work out how the virus enters and attaches to cells, it's extraordinary, the speed. But I think that breakthrough of these novel technologies will be important. I think the other thing that we're doing that is very different here is building the manufacturing factories without even knowing whether the vaccine works, and doing that at risk, because that delay, you know, if you wait until you know a vaccine works and then you start building, you know, the factories, that would really be pushing us into the 10-year time frame. And so there's a lot of risk there, and of the factories that are being built, you know, it isn't clear how many of them will actually produce vaccines that work. But I think that is a game changer for this pandemic. And it's a really important lesson for us to learn for subsequent pandemics, that this is a costly and risky undertaking, but nonetheless is absolutely essential to speed up the time.
Yeah, And you make a good point there about future pandemics.
Yes, unfortunately.
No, yes, unfortunately, that we know that we very likely face these threats going into the future. We've known for a while that perhaps on some level, in some way, I mean, that this was coming. It was interesting that Barney Graham from NIH, he was part of our first program, and for me, it was quite memorable when he explained that part of the reason that they've been able to move so quickly is that they'd already had this experience and this research investment in coronaviruses, like MERS, and made discoveries, and were well positioned to deal with another coronavirus versus one of another family. But at the Gates Foundation, I mean, clearly that you were able to move so quickly in 6 months, you were prepared for this, right? And I have to imagine that you remain even more prepared moving forward, having been through this process.
I think if you think about the fact that, we work on vaccines for HIV, TB, and malaria as our 3 biggest vaccine potential targets, we're talking in every year that those those diseases kill millions of people. And so the urgency that we see around those actually is shared with this type of urgency. So, building the toolkits that will allow us to build those vaccines as quickly as possible is really what we lent on here to respond to coronavirus. Clearly, this is a little bit more urgent.
Urgency is the one word for it, certainly one term that is appropriate here. And actually, with that, we've got a number of urgent, I think, questions from our audience. A lot of interest in what you've said, and some folks would like to know a bit more. Felix Guo, for example, he says, I've heard from news networks that the vaccine will be available by the end of the year. A timeline that is rather optimistic compared to the timeline that you've laid out, Lynda. Where in the vaccine development process is this big discrepancy or uncertainty coming from?
I think there's a difference between when we say available versus when it may be approved. So indeed, a vaccine may be approved early next year, but when I think about availability, and this is how I'm interpreting this question, it is when it's available for the shot in the arm of the people who need it. And if you think about it, to vaccinate the whole of America would be 300 million doses. To vaccinate the whole of India, 1.4 billion, China, 1.3 billion. There are a lot of people. So availability to the globe is a very different prospect than just when the vaccine is approved. So I believe that they would probably, I think you have to factor into how many doses would be available and who would be able to get those doses. And I think really, realistically, in any meaningful way, we're talking a year from now before there are available doses to protect the most vulnerable, even in America, and certainly not in the rest of the world.
We have a question here from David about trialing. Does phase 3 include children? And if not, can we confidently use the vaccine on children?
Yeah. So I actually do not know whether, so every vaccine developer will have a different plan for how they're going to run those studies. And some of those studies will have special populations that they will seek to protect. I think a major focus here is really looking at the elderly first and foremost. And so they're really skewing more to the elderly. But some developers will come down in age and will do an age de-escalation, as it's called, to see whether the vaccine is safe and immunogenic in children as well. I think it's really going to be a vaccine case-by-case basis to see whether, I ultimately think with the sort of description of this multi-inflammatory syndrome in children, that we are going to want a vaccine that protects children as well, clearly. And that's something we should be really looking at. The other populations that I think will need to be protected are actually pregnant women who are often excluded from vaccine studies. And if vaccine developers don't consider the reproductive toxicities, you have to do special testing to show that a vaccine is safe in pregnant women. So that's the other population that I think we're going to have to think very hard about how we reach and how we protect.
Incidentally, with a disease like COVID-19 that causes severe illness, and particularly the elderly, the age is a risk factor for those symptoms. How do you vaccinate a group that sometimes can't be vaccinated? That the people that need it the most might not even be able to have a vaccine. Is that right?
Yeah, so the elderly often do not respond well and robustly to vaccines, and we know that, for instance, for influenza, they often have very poor vaccine immune responses, and this is something people are concerned about, and this is actually why, for instance, antibody therapies may be really important, because you might be able to, instead of vaccinating, you might be able to give people protective antibodies that would cover them. So the elderly are a population we are concerned about seeing how these vaccines perform.
Okay, a lot to think about. I don't envy that. We got a question here from Niels. Is there a plan and consensus about the order of providing vaccines to protect the most vulnerable and also interrupt the spread of COVID-19? Has there been a standard developed, and does it include medical and ethical perspectives?
That's a fantastic question. And it's a question that I think is going to be a sort of ongoing debate for some time. There have been attempts to look at this by different agencies. For instance, the WHO is looking actively at this. And you can think of the most vulnerable in different ways. You can think of the most vulnerable as the elderly, who are the most at risk for severe disease. You could also think of the most vulnerable as healthcare workers who are on the front line, but who may not be necessarily of the age group that are high risk, and who I think every society will define this most vulnerable population slightly differently. So, as an example, in Italy, when the outbreak happened in Italy, Italy has a very high percentage of elderly in northern Italy, particularly. A lot of their population was over the age of 70 or 80. This is very different from somewhere like sub-Saharan Africa, where actually the population demographics is much younger. There are many fewer elderly, but in sub-Saharan Africa, there are different at-risk characteristics, such as, for instance, we don't fully know how this is going to play out, but for instance, HIV. So how does one define the most vulnerable is actually a ongoing debate. And then once we've defined that, how do we work out how to prioritize the most vulnerable is, I think, the second very complex problem that we face. Yeah,
that's really interesting. I mean, I've often thought about the term vulnerable being, something not so simple, but yeah, it's a really interesting issue. So we have another question here related to this about just trying to assess vulnerability, but also safety. Big concern about safety, right? When we're talking about speed. After a vaccine becomes available, would it be prudent to wait to see if there are unanticipated side effects? And if so, for how long? How long do we wait until we're certain that nothing is going to show up or cause problems down the road?
Yeah. Yeah, that's another great question. So I think when we think about vaccine safety, there are lots of different ways of thinking about it. There's is it safe, as in the first time you get it in your arm, does anything happen to you? And so that's something we evaluate in the phase 1 and phase 2 studies, right? They're designed to look for those immediate effects of being vaccinated. In the phase 2 and phase 3 studies, we look for the safety of how does the vaccine affect if you are exposed to the coronavirus, right? So if you've had the vaccine, do you do better or do you do worse? And so those are things we consider in the phase 3. There is also a phase of vaccine of that vaccine process, which is post-market pharmacovigilance and looking to see for many, many years after the vaccine has been deployed, whether there are any other untoward effects. Sometimes we don't see effects until that late stage after the vaccine has been approved. It's very, very unusual because mostly the safety is picked up in these earlier stages of development, but it is possible. I think it then becomes really a risk-benefit ratio, right? And understanding, what are by the time it's approved by the regulatory authorities, who are only interested in showing that it is both safe and efficacious, and the FDA and the EMA, they will not compromise on that. And they've released very stringent guidelines actually showing that they will not compromise safety in any way. I think at that point, I would say it is safe as best of our knowledge, and it's really balancing, your risk tolerance as to whether you do or don't take the vaccine. But generally, if the regulators, if you can get through the regulators, then they're very hard people to get through.
Yeah. And it was interesting to learn last week in our previous program from Paul Offit and Walt Ornstein about the surveillance that's done in the systems that are set up. Track exactly these particular signals, potentially, of what might be an adverse effect. Okay, from Gabrielle, we've got a question here of, if you'd like to address this one, Lynda, what's being done to prepare for and counter growing resistance to vaccines, right? That's a definite issue that we have to think about. There are conspiracy theories circulating about Bill Gates, for example. If too many people refuse vaccination, doesn't that endanger herd immunity?
So vaccine hesitancy is a concern, and it's been a growing concern, for across the board. And I think it's in some cases, people, for instance, in the measles vaccine situation, people have never seen measles. They have no idea of the impact. They don't know anybody who's suffered with it. I think in that case, the risks and the benefits are harder to understand. I think in this situation, understanding seeing the impact that coronavirus has on society is very present and very immediate for people, and I hope that will persuade people that the benefits are significant. However, Gabrielle's entirely right in pointing out that if you fail to get good uptake of the vaccine, we will not reach herd immunity. We don't quite know what herd immunity will be for coronavirus. We think we need 60 to 70% protection to really bring the R0 down sufficient for it to be eliminated from society. But we don't know that yet. And so clearly, if there is a vaccine that only 10% of population take, it will remain endemic and will remain as a circulating threat for time to come. The reason this is a concern, in my opinion, this is an RNA virus, and RNA viruses mutate, and we already are seeing mutations in this virus. So we don't really want to have coronavirus with us for the rest of forever, if we can help it.
Right.
Much better to get rid of it.
Yeah. Well, thank you for that. Yeah, that's a big question, and one we'll be discussing further in our next program. Okay, we have. I think for me, I think this is quite interesting because I've wondered this myself. Should people who have confirmed, who are already confirmed to have been infected with SARS-CoV-2, should they also receive the vaccine, or would they be lower priority to receive one upon delivery and distribution?
Yeah, so I think to understand that we need to know something we don't know, and what we don't know is whether prior infection protects you from subsequent infection. We know with other coronaviruses, like the common cold coronaviruses, that actually immunity wanes after time. So even if you've been infected, you're infectable a second time. And so, I would say at this point in time, I would err on the side of caution and say, even if you've been infected, that does not mean that you are protected from getting it again until we prove that. And in that regard, I would not change a prioritization for a vaccine based on prior infection or not, personally.
Okay. And we just got a question that I think continues on that point about immunity and people having it or not. Mark asks, he says, there were some stories about the MMR vaccine providing some level of immunity against COVID-19. I've not seen this. Is there any validity to that? And has any research been done to study that correlation?
So that's a great question. So there are a couple of things called non-specific effects of vaccines. This has actually been probably best articulated in the context of BCG vaccine, the tuberculosis vaccine that is given, where what you can show if you give people the BCG vaccine is that they are protected from all-cause mortality. So not just from TB, they're protected from other diseases, and it's significant. It's about 20% protection from other diseases. So, and similar things have also been reported in the MMR vaccine. So, in fact, there are ongoing studies in looking at BCG to actually ask, if you use BCG just to boost the immune system generally, does that actually protect you from severe COVID-19? Those are studies that are ongoing in the Netherlands, in India, and in Australia at the moment. And similar reports have also come out from MMR. And I think these often are not very strong effects, and therefore require really large studies to actually prove one way or the other.
Okay.
Possible.
Well, thank you. We've got time for one more question, unfortunately, but it is a good one. Actually, it touches on something that you just mentioned, and I think that a lot of people are wondering about. As the coronavirus mutates, as you just said, we now know that it is mutating, has done. How will a vaccine designed after researching the previous strain ensure protection?
So it's a really great question. Most of the vaccines are targeting this spike protein that's on the outside. And so although there are mutations in that, they are probably not major shifts. So unlike flu, which completely rearranges its spike protein, its hemoglobin, coronaviruses don't completely resort and do really funky things with that spike protein. As yet, at least, we don't think they do. And so the vaccines may become less efficacious and be less well matched with the circulating strains, but my hope is that they will remain able to protect because they cover that spike protein with antibodies. Single therapies, so single antibody therapies, are more risky of having escape variants that will no longer which will render them no longer effective, but less so for a vaccine. But it is a risk, and it's why we don't really want it to circulate a huge amount. Every time it gets into a person, it has an opportunity to do something new with itself, and so the less circulating virus, the less opportunity it has to vary.
That's a great point. It's like you're on it. You're aware. Yes. Well, Lynda, thank you so much. Truly, you provide so much insight into some really complicated and important issues, and to give us your time when you're doing so much, such important work. We're grateful. Really
Thank
great
you.
to have you join us.
Yeah, and it's great that the Smithsonian series really, both the fact that this existed prior to the COVID-19 was prescient, and I think keeping us connected to the science now is really important. So thank you very much.
Oh, well, we're doing our best, and only with support from folks like you. So thanks again. So to our audience, I will also say thank you for joining us. Thanks for your interest. I hope that you've learned a lot, and I hope that you'll come back and join us next week for the final webinar in this series, the fourth one, Vaccines and Society. Our speaker will be Dr. Sharon Kaufman, Professor Emeritus and former Chair of Anthropology, one of my people, History and Social Medicine at the University of California, San Francisco. We're going to discuss the policy and societal conditions driving vaccine hesitancy. So you can register for the webinar on the NMNH events page that will be shared in the Q&A box now. And please take the survey that will pop up once you leave the Zoom webinar. It's going to help us get feedback and continue to provide to you the programming that we think is so important. And to all of you, thanks again for your participation. Stay safe, be healthy, and take care. Bye.